Good afternoon, everyone. I think we're having slight technical difficulties as I'm unable to share my video, and I'm happy to go audio only, and maybe I will do that just so we sort of keep the show rolling. So first of all, to all of you friends, alumni, students, and colleagues, I wanted to welcome you to this special Mason Science Series this afternoon. This afternoon's Dean's Research Discussion. I am Fernando Morales Wilhelm, and it's my honor to serve as Dean of the Mason College of Science since July 1st, going on my 5th month now. I'm especially honored to host these events highlighting the extraordinary research that we have underway in our college. This semester, we've already had some really fascinating discussions around groundbreaking approaches to breast cancer treatment, forensic science program. And this afternoon, I'll have the pleasure of introducing an internationally recognized case study for ecosystem recovery. Well, it looks like my technical problems were solved, so here I am again on video. You know, more than 30 years ago, my own awareness and growing up in a increasingly aware environmental world inspired me to become a scientist. For the past 3 decades, I've worked as a hydrologist and water resources engineer. So naturally, this afternoon's topic is actually very special to me. But I've also learned that you know, that the more and more as my career has progressed, as I've worked with scientists in multiple disciplines, is that, you know, these disciplines are connected, and it is important to connect scientific disciplines if we're going to solve the grand challenges that we have as a society and as a planet. And I think our speaker today is a really great example, a true pioneer in convening scientists from various disciplines to solve the world's big problems. Now, before I introduced him, I wanted to go over a few Zoom housekeeping comments. We will record this event, and a link will be shared and posted on our website after the event. Questions can be submitted using your name or anonymous at any time using the Q&A box, and you'll find that in the bottom panel of Zoom. You'll find a little Q&A icon, so you can click there. And we will disable, you know, microphones, cameras, chat and raise hand features. So please interact through the Q&A box. So with that said, it's my pleasure to introduce my friend and distinguished colleague, Dr. Chris Jones. Dr. Jones joined our Mason Biology Department 40 years ago, back in 1980. In 2000, Chris was the founding chair of the Department of Environmental Science and Policy, which is home to the world's first doctoral program focused on the interaction of the environment and public policy. In 2007, Chris founded the Potomac Environmental Research and Education Center at Mason's beautiful Potomac Science Center in Woodbridge. This center brings together scientists from a wide variety of disciplines and departments to address sustainability of local ecosystems. It's a beautiful location with great facilities and an incredible team of scientists working there. And Chris has been the director of that campus of Mason for a while. He earned his PhD from the University of Wisconsin-Madison in botany, specifically in limnology and oceanography as a minor. He's a freshwater ecologist who specializes in the tidal freshwater ecosystems and stream ecology. He has published in a wide variety of journals, including Ecology, Estuaries and Coasts, Aquatic Botany, Hydrobiologia, the Journal of Ecology, among many others. He teaches classes in waterscape ecology, freshwater ecology, and multivariate analysis. He has supervised 23 PhD students in their dissertations and 39 master's students in their research. So Chris is a real scientist, a teacher, and his impact, his impact is intergenerational. He's been doing this for a long time. He's got a great legacy. This afternoon, Chris will discuss his research in Gunston Cove and Hunt Creek in the tidal Potomac that have resulted in a rare environmental success history. This research has been especially impactful due to the close collaboration with local government agencies who have worked with him since 1984 to integrate research findings with evolving practices in wastewater treatment, management of runoff, and it's really a very long record of how an ecosystem recovers. There are not that many success stories like this in the world. There are not many of these success stories that have actually been documented, like the work that Chris has done. So it's I also want to acknowledge that Chris, in addition to his research, his teaching activity, which is fantastic, is also a very generous donor to the College of Science. He has established an endowment to support environmental education activities. He generously supports a variety of programs across the college and across the university. Many of our alumni really may recall his rousing delivery of our national anthem at commencement ceremonies over the years. Thank you, Chris, for your generosity and spirit of support. So please join me, everyone, in giving Dr. Jones a warm welcome, a true Mason patriot.
Thank you very much, Dean Morales-Wilhelm. I'm so glad to have you on board here at the College of Science. I'm here today to tell you a story. It's a story of the remarkable recovery of our beloved Potomac River, and it shows what Americans can do to overcome serious environmental problems when they commit to solving them. So could I have the next slide? To set the scene for you, of course, the Chesapeake Bay is our kind of our mother water body, our mother estuary in this part of the world. And the Potomac River is tidal and therefore part of the Chesapeake Bay all the way up to Chain Bridge, all the way up to Washington, D.C. One thing I want to point out is that the upper part of the tidal zone contained in the red box is actually fresh water. So it's more like a lake than like the ocean. And so it's, that's because of all the fresh water coming down the flowing part of the Potomac River. There's 30,000 square kilometers of land up into West Virginia that drain down into this bay, down into this tidal river. And so we get about 50 kilometers in length of fresh water to work in. I just want to point you out to the lower middle part of the, within the red box is Gunston Cove, a Y-shaped body of water, about 20 kilometers downstream from Woodrow Wilson Bridge. Next slide. Okay, the original ecosystem of the tidal Potomac, we think, looks something like this in cross-section, that we had a deep water channel, and that deep water channel had a lot of oxygen in it, and it had healthy levels of algae, but not excessive levels. And then we had shallow areas along the flanks, and those contained a lot of aquatic macrophytes. We call them SAV, submerged aquatic vegetation. And this ecosystem supported a wide variety of organisms, fishes, invertebrates, mussels, and there were a lot of habitats for them to hang out in amongst the weeds or out in the fresh water. Next slide. But over the years, as humans populated the area along the river and had to dispose of their waste somewhere, the most obvious place to put it was in the river. And even if, and over the years, a series of different forms of treatment when it were instituted mainly directed at getting the oxygen consuming waste out of the river. But what remained was the nutrients. And the nutrients, particularly phosphorus in fresh water, kept coming in. They weren't being removed from the sewage. And as they built up, they caused algal blooms. And those algal blooms that are shown as these green dots in the upper part of the water column became massive. And you'll see pictures in a minute. They were enough to shade out the plant. So you see the little, much reduced SAV there on the left. And then when they settled to the bottom and consumed oxygen, they depleted the oxygen from the water. So the ecosystem had shifted to a degraded state by 1965. Next slide, please. Okay, and this shows you what these massive algal growths look like. You can see on the lower right, there's a photomicrogram that shows the individual cells in these cyanobacterial colonies. And you can see the massive amounts, look like somebody poured green paint on top of the water here from 1983. And that was due to, look at the table at the top, the massive increase in phosphorus loading into the tidal Potomac. Next slide. But the nation was awakening to the problems. In the mid-60s, there came a new awareness of the environment. And a major national and multi-state effort was initiated to clean up the nation's river. No less a personage than the President of the United States made a commitment to clean up the river. And to quote him, this river, rich in history and memory, which flows by our nation's capital, should serve as a model of scenic and recreational values for the entire country. So President Johnson made a commitment. And so the federal government got to work, and the Federal Water Pollution Control Agency, which soon became the EPA, began extensive studies to understand the problems, so with the goal of restoring the river to health. And this paper describes the response of one portion of the tidal Potomac, Gunston Cove, to this major initiative. Next slide. So here's another picture, another little map of the river. And what I want to point out is those circles are sewage treatment plants. And the bigger the circle, the more sewage being dumped in. Now, at this point in time, there was secondary treatment of sewage. So it's not like it's raw sewage, but it's sewage that had not had the nutrients removed from it. And so Blue Plains, the BP, is right there at the Wilson Bridge, is the largest plant. But there is one that's closer to us in part of Fairfax County called Noman Coal, and it dumps right into Gunston Cove. So that's the one we're going to study in this paper. Next slide. Now, so mid-60s and through the 70s, a lot of studies were done. The conclusion was, yes, there's too much phosphorus going into the river. We've got to reduce the amount of phosphorus going in. They geared up, spent a lot of money, millions of dollars, to put phosphorus removal technology into the treatment plants. And by the time I arrived on the scene, you can see even in this purple squares show the amount of phosphorus load going in. And by the early 80s, it was starting to plummet. They were getting the job done. They were taking the phosphorus out before the wastewater was discharged. So keep that in mind. By 1985, phosphorus loading was much reduced, 90% reduced. But how did the river respond? Next slide. Okay, so that was the question. And so I came onto the scene about 1984. The citizens that lived on Mason Neck, near the Noman Coal treatment plant, were becoming increasingly concerned about algal blooms and degradation in Gunston Cove. And they basically reached out to me and my colleague, Don Kelso, to ask us to put together a monitoring program so that they could establish the extent of the problem and recommend solutions, and then follow the course of those solutions. Very important. Next slide. So we started this in 1984. We went out twice a month during the growing season, basically April through September, and we sampled. We did a lot of sampling at different places for the first few years, and then we figured out that we only needed to sample at one sort of sentinel station, and that's what I'm going to show you is the results at that sentinel station. So I'm going to show you a little bit of some data here. It's pretty easy data to understand, though. It's just a scatter plot each year. I'll put the numbers that we got for a given variable, do some smoothing function, and look at linear regression to see if things were consistently going up or down. Next slide, please. So this shows total phosphorus, and each one of those circles is a measurement that we made. And you can see that, again, phosphorus is the limiting nutrient. And so phosphorus decreased, showed a strong decrease through after the cutoff of input phosphorus, but it didn't drop precipitously. It just slowly, gradually declined, and it's still declining some today. And so what's happened is that there was a lot of phosphorus sitting in the sediments, and even though they turned off the phosphorus spigot on the turned off the phosphorus spigot on the treatment plants, there was phosphorus that could well up from the sediments and kind of kept the levels up a little higher than they would. So we had to, that's the whole point of the long-term study. Things didn't happen immediately. Next slide. So gradual decline in phosphorus. Chlorophyll is a measure of the amount of those pesky algae in the water column who are absorbing light and keeping the plants, the macrophytes from growing. And you see that they were even less responsive. So from almost all the way to 2000, they didn't really change much at all, kind of hovering around 100 micrograms per liter, which is pretty green water if you see it. But eventually around 2000, they started to decline. So the phosphorus levels were titering down enough that now they were starting to inhibit the growth of the algae. So again, another lag in the system. And another important thing was that there was a shift in the dominant taxa from a cyanobacterium, which is considered a harmful algal bloom species, to diatoms, which are very much regarded as a good food source for the other critters in the river. Next slide. Okay, so with reduced phytoplankton, will SAV return? That's the question. So if we're trying to restore Gunston Cove, it's a shallow bay, it needs to have submersed macrophyte beds. The primary requirement for this is light availability throughout the water column. Now, things that absorb the light in the water column are the algae, inorganic particles, and dissolved substances. We don't have to worry too much about dissolved substances in the Potomac, so it's mainly algae and what you might call sediment. And in order for SAV to become established at a certain location, the sediment surface must receive at least 10% of surface light. Next slide. A simple way to measure water transparency is the Secchi disk. And on the left inset there, you see that is a Secchi disk. It is simply a flat circle made of plastic or metal and painted black and white in alternating quarters. And you basically lower that down until it just disappears. And that's a crude measure of, but accurate measure of water transparency. And so this shows the patterns of that. And that is the important thing for SAV, the macrophytes, are they getting enough light? And what you see is that values up until 2000, again, hovered at around less than 50 centimeters, less than half a meter. But after 2000, they gradually increased. And we had even some super clear days where we could actually see the bottom of the cove. Those are the circled ones there. So this increase in transparency goes hand in hand with decreased phytoplankton and should result in increased SAV. So to make a predictive model, we needed to know how the increased light we're seeing related to the 10% light threshold. Next slide. Okay, so what we saw was that at about the year 2000, so before 2000, the depth of 10% surface light was about 0.6 of a meter. And the tide range is about 0.6 of a meter. So there really wasn't much room for the plants to colonize. But by 2005, it had shifted to over a meter. And so we expected to see, starting about 2005, a regrowth of SAV in the cove. Next slide. So this shows the 2000 distribution of SAV, and we've circled Gunston Cove there, and the only places that SAV were found, even as late as 2000, were way up in these little bays called Occoquan Bay and Potomac Bay. Nothing out in the Gunston Cove itself. So but that was still the time when the predicted depth was only about 0.6 of a meter. So move forward to 2005, next slide. And you see a much larger portion now out into Gunston Cove is now covered with SAV. So the green shaded areas have aquatic plant growth. So it may not seem like much, but basically you went from very minor areas to half the Gunston Cove covered with these SAV plants, meaning that the habitat and the quality of the water, the quality of the habitat in that area has increased dramatically. Next slide. And 2015, one of our more recent times, you can see that it's maintained. Now, one thing I was hoping to see was that it would move even further out into the cove. So I have generally called this a partial recovery. Still amazing, but we're hoping that it can spread even further. Next slide. One of the things that's interesting to do is to show that this system seems to have had two stable states. In other words, the lower left, we have low maximum SAV colonization depth and low SAV area. So all the way through the 90s, I didn't plot all the 80s on here, but they would be in that red circle as well. And then you have an intermediate year, 04, and then you have a whole cluster since then, and I don't have all the dates since then, but where you have maximum SAV colonization depth of higher, much higher, and a lot more SAV. So it seems like within a period of 1 or 2 years, there was a strong shift in the cove toward more SAV, and it seems to be a new stable state, and this has continued. So we have a recovery. Next slide, and we can look, maybe easier to look at it in this type of graph. So the important thing is light. So that is the dark circles, and you can see that starting back in 1994, the light, the Secchi depth has increased quite consistently. There have been years when it's been off, like 2018 was a particularly bad year. There's no data for SAV that year, but it doesn't mean there was no SAV, but because they weren't able to fly, they weren't able to do collect the data. But it was a bad year. And it was also not a good year for, it's also not a chlorophyll was not up either. So chlorophyll is the red line. That's the algae that for most years are the main light blockers. But in 2018, it was inorganic sediment that was the main light blocker. But at any rate, you can see that you went from a very low level of SAV coverage of less than 100 hectares consistently to a much higher level of about 200 hectares after 2005, and it happened real fast. So we'd like to see further increases in that. Next slide. So we've documented the partial restoration of Gunston Cove, including decrease in pea loading, decrease in phytoplankton, increase in water clarity, reestablishment of SAV beds, and an apparent regime shift from phytoplankton dominance to partial SAV dominance. Next slide. As far as this research goes, this has definitely been a cooperative agreement between the county's Fairfax County Department of Public Works and George Mason. There's been a lot of in-kind contributions on the part of both. So they regard it and we regard it as a major success story. I just published in an international journal this full study, and it's available on our PEREC website. I published it at open access, so anyone can get it. Okay, next slide. Okay, so basically that's what I wanted to present about my research. And then I was also invited to talk a little bit about our new center at Belmont Bay. So we're switching presentations here. I was also encouraged to take my camera and prove to you that I'm actually out overlooking the river here. So I'm down at my home base. Okay, so some of you may know about this, but in 2018, we opened the Potomac Science Center. The main client there occupant is the Potomac Environmental Research and Education Center. We are part of the College of Science. Next slide. Just to briefly, our mission is to use the tools of scientific research, restoration, education, and policy analysis to help society understand, sustain natural processes in ecosystems, watersheds, and landscapes through the typical university routes of research and scholarship, instruction, and outreach and events. Next slide. You can see where we're located right at the end of 123, essentially. If you drive down 123 all the way from Fairfax, all you need to do is hop over the railroad track and you're here. Of course, that's a little harder than it sounds. Next slide. So this gives you a little bit better idea of some of and you're welcome to come down. There's a trail that runs right around our building, and the building's open from 9 to 5 daily. We don't really have a whole lot to show you right now, but it's a great place just to look at the river and enjoy. Next slide. Okay, just to briefly cover our facilities, we have 8 faculty research wet labs. We have 1 teaching wet lab. We have a whole lot of support rooms with specialized equipment. We have a large multi-purpose event room, which is rentable by the public, and it has a huge view of the river. We have a K-12 discovery lab, which we're still trying to network with local schools to make that a big asset. And we have offices, of course, for us here. Next slide. So just wanted to give you a few nice pictures to encourage you. On the left, you can see the view from the patio out over the river. Below that is the view from the lecture room. It's kind kind of hard to concentrate in that lecture room sometimes. You have a plant wall off the upper right, and then we actually have a pond as an additional aquatic feature. Then we have a nice platform we can do sampling off of that pond. So what are we lacking? Next slide. One thing we're lacking is a pier, which will allow us to dock boats at the center. It's pretty shallow right off the center, and we actually need to run about 500 feet out to reach the channel. And we have recently received approval for a fundraising project for something called the Don Kelso Learning Pier. So this would be in honor. He's still around. He's not at Mason anymore, but he's still kicking. So this would be in honor of my colleague, Dr. Kelso. And this would allow us to really project it out into the river from the center. Right now, we often have to trailer our boats to a launch ramp. Next slide. This is a list of the faculty here, and obviously can't run through all of them. You can briefly look at their research areas, and also note, as the dean pointed out, that this is an interdisciplinary group. We've got the departments are shown in parentheses: ESP, chemistry, atmospheric, oceanic, and earth science. So we've got, we even have some engineers down there now. So it is very interdisciplinary. Next slide. This just quickly shows you where the Hunting Creek study is versus the Gunston Cove study versus some monitoring in Fairfax City, and also some work out at the Prince William landfill. Next slide. So our other major study is Hunting Creek. We've started that in 2013, to track the effectiveness of remedial measures to control nutrient loading in the Potomac. One of the issues that they have in Alexandria is combined sewers, which means there are certain parts of the city where the toilet waste goes into the same set of pipes as the road runoff. Obviously, that's an issue, and it's being remediated, but slowly. We've been looking at pharmaceuticals in the water, and doing water quality mapping in partnership with Alexandria Renew Enterprises, which is the Alexandria Sewer Authority, basically. Next slide. And one of the interesting things we came up with at Hunting Creek was that you probably have driven right along Cameron Run. Cameron Run is the stream that the Beltway runs along from Old Town Alexandria out toward Springfield. You look over there, there's a creek, a river, and it's just considered this urban, collection channel. And to everyone's surprise, we've discovered river herring spawning in Cameron Run. And so that's, one of those things where the fish are they're pretty resilient, and you just have to go look for them. Next slide. Some of the other areas of active research, microplastics, which has certainly come to the fore, both in and of themselves and also as carriers of pollution. Harmful algal blooms. You see that garden rake on the lower right is hosting a mass of cyanobacteria, and we're investigating their toxicity now. Invasive freshwater and marine organisms, coral disease outbreaks, and some really interesting work on crab parasites, which would require more time than I have to talk about today. Next slide. So I just wanted to mention in particular that we have a very robust outreach program for 6th and 7th graders in the 2 large local school systems. And every year, except right now, we provided outdoor experiences for 11,000 students locally. And it also serves as a training for university students to serve as field interpreters. And that's Cindy Smith, who's the director of that program. Next slide. So that's my talk. on the in the upper right, you'll see the kind of pier we'd like to be able to pull our boat into a pier like that and walk up to the building. So that's there for purpose. But we're really looking forward to great things, and particularly under the leadership of our new dean, we think we have much to offer and much to do. So thank you very much.
Thank you, Chris, for very informative, very relevant presentation, as well as for that update on the Potomac Science Center and the facilities and all the great work that you're doing out there. We do have a few questions on the queue, so I'm gonna read them to you, and I'll let you answer them. First one: has the phosphorus level of input gone down further in more recent years? And how has the politics affected the amount of phosphorus and nitrogen levels found in the Potomac? So that's one question.
I see the last little piece of that too. Yes, well, actually, the phosphorus levels of input from the treatment plants have not gone down much below where you saw them, but the volume of waste has increased. So they've actually continued to reduce the concentration of phosphorus in the wastewater. Now nitrogen was another problem that was different. It took them another 15 to 20 years to get that figured out. And I would say that the local governments in the DC area partially because the citizens want it, have gone to the limits as far as cleaning up the sewage, and they are at state-of-the-art limits as far as cleaning up the sewage. And it hasn't really, they haven't changed their game, no matter who's been in the White House. So that's not been a problem. I mean, the large cities are actually doing an incredibly good job of keeping waste out of the bay.
Great. So what do you recommend to do to combat algae blooms?
Reduce nitrogen and phosphorus input. So if they've got enough nitrogen and phosphorus to grow, they will grow. Now you can try, people over the years have tried to poison them or various things like that, and it's a losing game. So you have to control the source of the food for the blooms.
Okay. So another question, I presume the primary source of water for Gunston Cove is the Potomac. The SAB recovery areas are in the upper area of the cove, farthest away from the main Potomac flow. Does this mean the recovery is dependent more on the health of the upper tributaries than the Potomac itself?
Yes, so the primary source of water for Gunston Cove is the tributary of Occoquan and Pohick Creek, and also especially the sewage treatment plant effluent during certain times of the year is one of the main tributaries. But it does interact with the water from the Potomac. Yeah, so I think the answer is it depends on all of the flow from the river and the tributaries, and all of those, like Blue Plains, all of those have reduced phosphorus down to pretty minuscule levels. So everyone's working together, I guess is the best way to say it.
Okay. Do you work with Professor Amy Fowler's class? I was in her class in spring 2019 and saw the area.
Yeah, well, Dr. Fowler is one of our co-PIs on the grant. Mm-hmm. So she specializes in the benthic organisms that live in the river and how they have responded to less nutrients. Yeah, so I work with her. I mean, I don't spend a lot of time in her classes, but I have worked with them some.
Yep. Here's a comment from it's one of our alumni indicating that Dr. Kelso was his ecology professor back in 1973. Yep. We do have a legacy. That's awesome.
Yeah. Yes, I would say that's he definitely, it was true.
So is hydrilla a sign of clean water or a problem? Can it be controlled, or is this a positive change?
So the hydrilla story is really interesting because while it didn't, certain parts of the Potomac, like up around Wilson Bridge, actually got SAV back much sooner than Gunston Cove did. And the SAV that they got was hydrilla. And it was leaked out of a pond in Constitution Garden. And they didn't think it would live in our habitat, but it did. And so there was a lot of concern because it grew very lushly and it blocked boats, you know, it made it hard to get boats out. But what happened was, as it cleared the water, as it stilled the water and the sediments came out of the water, native plants started appearing. So the hydrilla was actually the leading edge of the recolonization of SAV into the Potomac River. So yeah, it's still very common. So in a sense, yes, it is not native, but it allowed the natives to get back in. And we have now a lot more natives. So I would say it was a good thing.
Okay. Yeah, to what extent does the health of the land-based ecosystems along the river shore affect the health of the river? Do robust ecosystems onshore help substantially with the river health?
Well, I think the health of the land-based ecosystems is very important, and not just along the river shore, but all through the watersheds that drain into the river. Because now that we've been able to get the phosphorus under control and the algae, we're finding probably the reason why we haven't had more and even greater growth of the SAV is erosion of suspended sediment from the watershed. And it appears that that is now the main blockage of light is coming from those. So I think we need to have a whole watershed approach to having the healthiest Potomac we can.
Okay. Can you comment on why the NC plant discharge has not increased with increased population density?
Yes, because technology, toilets now have less capacity, and there have been other parts of houses that have lowered their water consumption, and it's basically a direct pass-through of water consumption. And like showers now have controllers on them, and you don't. So it's been part of our whole package of trying to be more water conservative, even though we don't live in California where there's severe water shortages. Technology has advanced. So you're dealing with more people, but the amount of flow of water hasn't increased.
Right, right. Are you measuring pesticides and herbicides or other urban runoff products?
So in our, we have not done that work in Gunston Cove. It does require a more specialized chemist than we've normally had with us. But when we started this study up at Hunting Creek, they were very interested in some of these organic pollutants. And we started with PCBs, and then more recently, we've been doing pharmaceuticals. So we're doing organic chemicals. We haven't hit pesticides and herbicides quite as much, but we are aware of those.
Okay. Here's an interesting comment. So there are a number of kayakers in Belmont Bay next to your centers. So we would appreciate it if you can have your dock elevated so we can go under your ramp or dock to the marsh next door. Any thoughts on that?
Okay, so I think they're referring to the proposed
right
pier, and yeah, I think that's a consideration that should be definitely thought of, definitely incorporated into our plan.
Good.
Yeah.
Do you know of any other places in the U.S. that are following the example of Gunston Cove and aiming to clean up their water discharge as well?
Well, I think all the major urban areas are under court orders or are voluntarily doing everything they can to remove nutrients from their effluent. I think what we've been able to do is have a consistent data record that basically proves, especially with the modeling that we did too, that this was a result of nutrient removal, and that there were lags in the system that a lot of times people will give up. They'll quit sampling it. Well, nothing's changed. I guess we're just going to quit sampling. So yeah, I mean, there are many places that are following this example.
Right. What other environmental problems are observed in the Potomac?
Well, okay, I had a slide there, about plastics. There's concern about plastics. There's concern about combined sewer overflows, which combined sewer overflows are the reason why you can't swim in the Potomac, because if you have any type of rainfall event within a week or two of when you might want to swim, it was bringing in raw sewage into the river. That is being solved or greatly remediated by some underground storage systems that are being built both in DC and in Alexandria. We have invasive species, snakeheads. Blue catfish is probably the most worrisome. That's capable of eating a lot of desirable species. So these are all things to watch out for. That's what I would say.
Okay. Besides crabs, are there other benthic microinvertebrate studies going on?
Well, we do monitoring, and of course, the crab work is mainly not done on site here because we're freshwater and the crabs like at least a little bit of salinity. Most of what's going on is looking at benthic macroinvertebrates in the tidal river. And one of the interesting things that we found is that the native mussels are still out there and they're actually proliferating. So there was a period of time in which it appeared that native mussels had been eradicated from the tidal Potomac due to sewage. And now we're starting to find them coming back in. So I think that's the best thing I can point to.
Okay. Let's see, is there any way for undergrad Mason students to help research at the center for help faculty with research at the center?
Mm-hmm. Okay, so there's we typically have a Oscar group project going on here, so you can apply for the Oscar research project at Potomac at PEREC. Otherwise, you could just approach individual faculty. It would probably be the best way to do it, and see if they had a spot for you in their lab.
Okay. What can individual citizens do to help the health of the Potomac?
Well, lobby local government to continue to do the best sewage treatment possible, to control development, and where there is development, make sure there is adequate treatment, adequate BMPs, best management practices put in place, and just kind of keep telling your politicians that you're watching them on that, I would say.
Good, good, good. If the tributaries are a primary source of Gunston Cove pollution and the tests are showing that phosphorus and nitrogen levels have decreased, that would seem to indicate there's been a significant success in cleaning the pollution. If this is the case, that is a major success story in this highly urbanized area. That was a comment.
Right. I agree. Thumbs up. Thank you.
Where's the majority of the phosphorus come from, and how did you measure those levels?
Okay. Well, it came from the food that you eat, basically. The food that you eat, and it doesn't all, we don't need as much phosphorus as there is in the food that we eat. So a lot of it will go out into our toilet, and that will get flushed down to the treatment plant. And then if it's not removed, it will cause these terrible problems. There is some phosphorus that would, non-point sources that would result from over-fertilizing your lawn, for example. Measure the levels. It is a I'm going to use a word, it's a lab chemistry test. There is no magic probe for phosphorus. You have to take a sample, digest it, do a protocol that takes the phosphorus and converts it into a colored compound, and you measure it in a spectrophotometer. And that's kind of old-fashioned lab benchtop chemistry.
Okay. I live near a creek that feeds into the Potomac River. It has undergone a restoration of the banks. Does this prevent more suspended sediments from entering the Potomac?
Well, that's their goal. So that gets into a whole nother controversy. But at any rate, one of the ways that so if you go into creeks in urban areas, you see that the creeks have downcut and are starting to erode their bank. And all of that soil that was under the bank ends up down in the river once it's washed. So the idea in stream restoration is to lay back those banks. So create, kind of say, accept the fact that you're going to have more water through there and build a wider channel. And then there's not as much sediment to come down. So yes, it should be healthy.
Okay. Can you project the health of the river 10 or 20 years out?
I can. I guess I could project that that it has now reached a new steady state that with a lot of SAV, and I hope that will continue. But yeah, there are unpredictable things that happen, and other pollutants that might come in. There's global climate change, so I can't project it too well.
Okay. What fish, if any, are safe or safest to eat from the Potomac?
I think as long as you don't eat them every day, you can eat almost any fish that tastes good. And the farther you are from DC, the less likely you are to have either bacteria or toxic chemicals in your fish. Okay. But of course, the fish swim around, so you don't know exactly where it's been.
Right, right.
But normally they say you should eat river fish like no more than once a week.
Yeah. Okay. Are you considering climate change in any of your studies?
Well, I'm always receptive to, I always look at that to see if, for example, we've got a gazillion measurements of water temperature over the 38 years. And there hasn't really been it's very close to a marginally significant increase, but nothing real strong. So we really don't have much to go on as far as climate change.
Okay. Is there any practical way to remove nutrients from the existing sediment?
I would say no, just because you could do it, but it's not practical in a large setting. I mean, a small pond, you could go in and just scoop out all the existing sediment, and that would take care of you to remove the nutrients too. But to try to parse them out from the rest of the sediment, it's really not practical.
Okay. Has Mason done any studies in the non-tidal Potomac, like upstream of Little Falls?
Not much. We've done some tributaries that come in to the tidal part. We've done quite a bit of work there, but we haven't done very much on the upstream of Little Falls area.
Where can we find out what mussels or fauna are in the Northern Virginia area of the Potomac? Are there any invertebrate guides or similar?
Okay, so I could look around and find. I can't point you to a single source. The Fairfax County Stream Survey has done a very good job of inventorying all the organisms in their streams, including mussels. So I would go to look at the Fairfax County Stream Survey data. There are invertebrate guides, but they're not really specific for this area. They're more generalized.
Okay, here's an interesting question. What is Mason doing in the city of Fairfax? This comes from a former student who did restoration work in Occoquan Creek in the mid-90s. After graduating at Mason, helping Dr. Kelso with monitoring in Gunston Cove.
Mm-hmm. Well, right now, we just do their quarterly MS4 monitoring. So we're doing a baseline monitoring. It's really not research, I would say. It's kind of a contract that they have to monitor every quarter. We're also looking at doing storm monitoring, high flow monitoring for them as well.
Okay. Do you measure stream volumes and flows to gauge how dilution affects loadings?
Well, we don't measure stream volumes and flows. So I've looked at loadings and I've looked at concentration, but I haven't done the math in between those two to see if it makes sense, if the dilution is consistent with that.
Okay. Yeah, there are a couple more questions in the chat that I'll read. So your study shows the value of long-term consistent monitoring to detect change in an ecosystem. How many studies like yours do we have in the Chesapeake Bay region?
Okay, well, I'm not aware of a study that as quite as long as ours and as consistent. But there are, I'd say, half a dozen other systems that have shorter monitoring periods, but have shown the effectiveness of phosphorus removal from sewage.
Okay. You have shown that Gunston Cove has recovered significantly, but not fully, from the impacts of eutrophication. What remains to be done for it to fully recover?
So, as I was saying, I think that the major source of turbidity, in other words, what's keeping the water from becoming even clearer, is suspended sediment from the urbanizing watersheds. that drain into it. So I think the algae problem has been solved, but until we get less inorganic solids coming in from erosion, we may not get a full restoration.
Great. Well, I think that closes it out in terms of questions. I want to thank you, Chris, for your talk, for your candidness and in answering these questions. I will note that in the chat, and I remember this was recorded, there are a couple of responses. For example, there's a field guide to Fairfax County's plants and wildlife. There's a little link there. Also, my colleague, Professor Karen First, says that he's a new professor at the Potomac Science Center focusing on water chemistry, and he will be looking for undergraduate researchers. So you can reach out to Professor First as well. So I want to thank everybody for attending. This was certainly a fun session, lots of interest. Certainly the topic was very timely. So thanks again, Chris. Thanks to the organizers. And I wish you a pleasant evening. Stay healthy. Stay tuned for next one. We'll be doing more of these. You'll be receiving a notice, and I look forward to continuing seeing you. Good evening, everybody.
Thank you.
Take care now.
Bye.
Bye.